Welcome to the Proteomics in Proximity podcast, where your co-hosts Cindy Lawley and Sarantis Clamidus from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Cindy and Sarantis.
Hey everyone, welcome to Proteomics in Proximity. Today we have some exciting guests. We've got Rory Collins from the UK Biobank and Chris Whelan from J&J. We also have my colleague from Olink, Evan Mills. I would really like each of you to introduce yourself, talk a little bit about your why, and maybe a little bit about why you're here today and what we plan to discuss. Rory, let's start with you.
Well, thanks very much, Cindy, for inviting me to talk about UK Biobank and this fantastic step forward in analyzing proteomics in UK Biobank. So fundamentally, I'm a cardiovascular epidemiologist and clinical trialist. I've been at the University of Oxford for the last 40-odd years. And back in 2005, I was asked by the Wellcome Trust and the Medical Research Council if I would take on the role of establishing UK Biobank. So this cohort of 500,000 British men and women who provided lots of information from questionnaires, allowed us to measure them in all sorts of ways, and to provide biological samples, in particular blood samples that we stored 20 years ago. We've then been following them up with their consent through linkage to all of their medical and other health-related records, and importantly, making all of these data available to scientists around the world, whether academic or commercial scientists, to try to understand the determinants of different diseases and better ways to prevent and treat those diseases. The samples have had biochemistry done, hematology done, genetics done on them, including sequencing. But what's happening now, I think, is a massive step forward. The ability to measure thousands of proteins on these very large numbers of individuals is going to be a huge improvement in our ability to understand how to better prevent and treat disease.
I love that. So, Chris, how about you? Background, and then I'd love it if you could talk about, J&J's perspective.
Great to be back, Cindy. Thanks for having me again. It's terrific to see Evan on the podcast this time as well. So, yes, I'm Chris Whelan. I lead the Pharma Proteomics project. I co-founded that consortium about 5 years ago now. Formerly, my PhD was in neuroscience and in genetics, but over the last few years, I've really transitioned and deep dived into proteomics. I think that deep dive was driven by a desire to understand human health and disease at a much finer grained level. Ultimately, I want to live in a world where we can directly monitor and detect and treat illnesses in a more powerful way than currently possible. And UK Biobank is enabling that.
And why do you see proteomics, this proteomics project that Rory referenced, where do you see this as beneficial to pharma, just at a very high level? Because I think we're going to dig into this more as along our discussion today. But I'd love just your why. Because it's taken a lot of work to put this together. There are, 13 pharma partners in the first project, the pilot of over 54,000 samples. And now there's 14 pharma partners in this latest iteration.
Sure thing. Yeah, absolutely. I mean, I'll sound like a broken record, Cindy, but I'm all about precision medicine, finding the right drug for the right patient at the right time. And I think proteomics will help us get there quicker than any other tools that are currently available. I think it's the key that unlocks precision medicine. So you need a lot of statistical power to do proteomics in a sort of solid manner. And I can't think of a better cohort in which to do a really well-powered study than UK Biobank. So, as we all know, we've just announced the latest iteration of the UKB PPP projects. So it was 13 partners last time around. It's 14 partners this time around. And last time it was about 55,000 samples. This time we're starting with 300,000 and we hope to expand that to 600,000 pending additional sources of funding.
Amazing. Super exciting. All right, Evan, you're on. Your why, your background, whatever you'd like to share.
Sure.
Thanks again, Cindy. It's nice to be back on the podcast. I know that I had a previous experience. So I've been with proteomics companies, and I would consider next generation proteomics companies for about 11 years. I started my career as a research scientist in neuroscience and oncology. But my goal was always to do something that could actually impact patients. So I moved into a pharmaceutical sales role, which was not very satisfying, to be frank, but I've been in the life science tools business for about 16 years. And my goal is to put the best tools in the hands of the best scientists on the planet to make meaningful change towards improving human health. And so fortunately, Chris and I had lunch one fateful day in Boston. I think it was probably, 5 years ago now. And Chris just asked the question. He said, "Hey, I'm on this UKB steering committee, and we're thinking of a phenotypic data arm. Like, what can we do after sequencing, right? We're doing whole exome. We're going to do whole genome. What do we do next?" And he said, "Do you think Olink could possibly measure 50,000 samples?" And at the time, it was a bit of a pipe dream, but I kind of knew what was coming, and I said, "I think we can." And so that began this beautiful process that brought us to where we are now, where I've been in the fortunate position of representing a technology that's really enabling quite a bit for the research community, and working with Rory and team, the combination of really game-changing tools with unique to the world resources, with people like Chris that have the passion and motivation to make things happen, has brought us to where we are. So I'm very fortunate and excited to see what comes after this.
Amazing. As an aside, I will say the episodes that Evan and Chris were on respectively are two of the episodes that I get the most inquiries on, that we get the most hits on. They're very popular episodes. In fact, somebody sent me an email saying that they wanted to work for Evan after his podcast episode. So you should go back.
Me blush.
We'll put a link to that episode and Chris's episode in the show notes, because those were very good episodes. The work that you all have really spearheaded and consolidated resources to do required money. And that money, in part, has come from pharmaceutical companies. In part, it's come from an investment in Olink. I think, Evan, you and I both know that that's been, a lot of a subject of internal conversations. We're really about advancing precision medicine here at Olink as well, and understanding diversity. And I think this next step will have a lot of diverse samples in it. 10 times the ones that they had in the first project. But when thinking about funding, certainly the UK government has been a big supporter of the UK Biobank. And so, Rory, in particular, if you're in an elevator and you need to talk to someone from the NHS or from Wellcome Trust or from one of the funding agencies where you're familiar with their goals, what is your pitch about why proteomics should be done on a large cohort with outcome data like the health records in the UK Biobank? What do you say in that elevator?
Well, the first thing I say is, why should they engage with UK Biobank? So what's so important about UK Biobank? It's not a research project. It's not a national resource. It's an international resource. It's something that's used by thousands of scientists around the world. It is the Hubble telescope or the CERN accelerator or biological science now. Industry and academia go to UK Biobank data because it allows them to do things that they couldn't otherwise do. And I think the UK government are proud to have been part of creating that through the Medical Research Council and obviously the Wellcome Trust charity. I think there are 2 points that one can make to them. By putting resource into UK Biobank, creating UK Biobank, they've leveraged enormous investment from external sources for the sequencing, for the proteomics now, for imaging of participants. So from a financial perspective, their investment is leveraging additional investment in a resource that is of value to UK scientists and global scientists. More importantly, I think, from all of our perspective, is they're leveraging better health. They're providing data that is allowing scientists around the world to work out better how to prevent and treat disease. And I think what's really important about the proteomics, as Chris has alluded to, is that in a way it's the common pathway. There's been lots of excitement over the last 10, 20 years around genetics. But the genetics lead to disease through a pathway. And that's a common pathway for lifestyle, environment, genetics, and other factors. And the proteins will be that common pathway. And I think that's why the analysis of proteomics, thousands of proteins, thousands of pathways to tell us how lifestyle, environment, genetics leads a particular individual to develop a particular disease. And that's where I think we're going to see massive knowledge generated, which will help us to work out how to better prevent and treat disease. And that would be my rather long elevator pitch, but I was in a tall building.
That's a long elevator ride. I will say you've created an environment where the sharing is controlled and managed and safe that welcomes international participants to feel comfortable interacting with those data. And that's, I think, a fundamental piece that I've seen that people really appreciate there. Chris, I'd love your thoughts on how you convince leadership within not just your company. How do you support those scientists that are representing other companies in speaking to their leadership about being in a part of this team effort, this consortium?
Well, somewhat ironically, for a proteomics consortium, we're actually predominantly populated by human geneticists. That's actually been a huge driver in convincing our leadership teams in the value of proteomics. We've been advocating for the last 5 to 6 years on the value of human genetics for drug discovery. I think we've all seen the papers and the presentations that have suggested that if your drug target has supporting evidence from human genetics, it's at least twice as likely to ultimately make it to the market or be approved by regulatory bodies. But there's still a lot of missing pieces between, the genetic variant and the actual disease phenotype. And I think the proteomics is increasingly being recognized as a tool that we can use to bridge that gap and help understand at a much more finer grained molecular level what's happening between that pathway between gene and disease phenotype. So there's growing traction, there's growing appreciation from our heads of R&D that this is a very important, potentially transformative new research tool.
Evan, any thoughts you want to share on any of that? I mean, you had to convince our internal leadership of the importance of this project. I don't think I think they came on board pretty quickly, but.
No, but it's a fair question, Cindy. I mean, to enable really transformative projects sometimes requires a commercial entity to take some risk. And I think that's exactly what happened here. The promise of proteomics is fairly clear just based on the central dogma and what you've heard from Rory and Chris. I mean, there's clear utility in looking at the proteins, but technological limitations and frankly cost have been significant barriers. So I have to give all credit to John Heimer, CEO of Olink, who really had the foresight to go to the board and push for something that was simply unheard of to enable the project. And I think we can't underscore the importance of companies that have a really powerful technology. Sometimes you have to put profits aside and just think about impact. And I think this is a really good example of that.
I love that. I think it's a technology I've described as a rising tide lifting all boats. And there are many of those technologies around for human health in the context of genetics. I've seen few that are, I'd say, punching above their weight and, over-delivering what I expected anyway. So I am very excited about things to come. I'd like to touch back on diversity in running the entire UK Biobank, for example, with whole exome data, whole sequencing data, whole genome sequencing data. Yeah, there's a large representation of African diaspora and South Asian ancestry that I just like people to realize. I just want to point it out because that's one of the things that I'm particularly excited about in the plans for running this larger next step in proteomics. So I wonder if any of you would like to make comments on that. Chris, I'll ask you first if that's all right, or we can go to you, Rory.
I'm certainly happy to comment on this. I mean, as an epidemiologist, I think of people being much more similar than dissimilar. So I find the focus on diversity a little bit odd in a way. We are all human. Blood pressure is strongly predictive of the risk of stroke in all ethnic groups. Blood cholesterol is strongly predictive of the risk of cardiovascular disease in all ethnic groups. The reason why, as an epidemiologist, one would want to measure cholesterol, for example, in different populations is that the levels are different in different populations. So it was really our work in China showing that very much lower levels of cholesterol than we see in the West were associated with very much lower rates of coronary artery disease that drove our studies in the UK to look at lowering cholesterol in people with so-called normal cholesterol levels and demonstrated that we could lower their risk. So the reason for thinking about studies in different settings is to be able to study a wider range of risk factor levels, or to study populations that have different levels of disease, higher rates or lower rates of disease. If you want to study cerebral hemorrhage, do your studies in China, not in the UK or in Western populations, because it's much more common there. And so what we need is not diversity so much in terms of ethnicity, but diversity in terms of risk exposures. The reason why I think that that's valuable from a genetic perspective is that there have been particular genetic variants, if you like, that have been in particular populations. And that makes it very valuable to be able to study genetics in different populations. But equally, as I say, for studying environmental or lifestyle in different populations. You're right that there will be quite a lot of diversity within UK Biobank, but not enough to really look at the full range of exposure levels and to look at the full range of disease levels. But in the same way that the first 50,000 participants in UK Biobank having proteomics is a pilot for doing it in the whole of UK Biobank, I see UK Biobank as being a pilot for doing proteomics in the other large-scale studies that have been established in other parts of the world, in Mexico, in China, in North America, particularly in Hispanic populations, in say, the All of Us study. So no one study answers all questions. I think what we're doing is building on the knowledge we have and then building on that knowledge. And that's why I think this is a very important next step in understanding the diversity of human disease.
I like how you flip that from thinking about diversity, which is a foundationally sort of genetic construct, to looking at representation of disease state, because that's where we're going to be able to understand more about proteins showing up in those disease states. And so representing, as much of an understanding in epidemiology as we can.
Yeah, the kind of minority of people from African backgrounds or Asian backgrounds in UK Biobank will not be the ones that tell us predominantly about the relevance of proteins to disease in Africa or Asia. It will be the totality of UK Biobank that will do that.
That's really helpful perspective.
So with this expansion project, I mean, as Rory said, we'll not only capture more samples from underrepresented populations, which is absolutely crucial, but we'll also capture, in my opinion, samples from underrepresented illnesses. So we're going to start by assaying 300,000 samples. 250,000 approximately of those samples will be from the baseline visit, and then an additional approximate 50,000 will be from various repeat assessments. As somebody who primarily works in neuroscience and in rare diseases, we were maybe somewhat underpowered to study certain diseases of interest in that pilot proteomics data set. Let's just take an illness like myasthenia gravis, right? That's an illness that I'm quite interested in, but we only had a couple dozen cases in that pilot project. We'll go from a couple dozen to hundreds of cases. Schizophrenia is another good example. I have a lot of interest in that. We had maybe 150 cases in the pilot. We'll go to maybe more than 2,000 cases in the full-scale project. So that will be a game changer for biomarker discovery. But it's also incredibly exciting because of those folks with repeat samples, there will be approximately up to 80,000, maybe up to 40,000 in that first 300K cohort, but ultimately up to 80,000 that will have plasma proteomics on samples that are collected contemporaneously with whole body MRI scans. So that will give us next level biological granularity. We can go from microscopic to macroscopic, and that didn't really exist in the pilot study. So you can imagine not just saying this blood protein is changed in people with bipolar disorder. You could say this blood protein associates with white matter microstructural alterations in the corpus callosum of people with bipolar disorder. It just gives a level of granularity that could really be game changing.
It feels like functional genomics. Like it just feels like we're getting, it doesn't answer all questions. It's corroborative perhaps with true methods of functional genomics. I just think there's so much potential. Chris, would you be willing to talk to us a little bit about how bringing, you mentioned genetics helps us, I think there's a Matt Nelson paper on this, there's a couple of other publications around how genetics helps build confidence in clinical trial success. How does bringing proteins, genetics, and clinical outcome data like we have in the UK Biobank, how does that help your company or a pharma company in general have more confidence in the therapeutic targets that they're building molecules for?
So there's a multitude of ways that we're using these kinds of data. I think the lowest hanging fruit, as you pointed out, Cindy, is specifically for target discovery. We mentioned earlier the increased confidence in drug targets that have supporting evidence from human genetics. What the protein data allows to do when they're combined with genomics is actually pinpoint the proteins that we should be targeting. Obviously, most of the drugs that we develop are targeting proteins, they're not targeting genes. So just finding the gene that's linked to your disease and having high confidence that in the gene linked to your disease doesn't get you all the way. Ultimately, you need to figure out which protein has a causal link to disease. So we employ techniques like Mendelian randomization that help identify or establish that causal association with disease. And we've done this across the board for countless disease areas. The example that I often point to, because it's my team at J&J who did a lot of the work, is Parkinson's disease. We did some proteogenomic modeling. We identified dozens of new targets for Parkinson's disease that weren't previously identified using traditional GWAS. So galectin-3 is a good example there. We published on that recently in Nature Comms. But we've also identified inflammatory targets for schizophrenia and Alzheimer's disease and a variety of other conditions. I would say that one of the things I'm most excited about in terms of the applications of proteomics in the context of pharma is how we're applying AI on the protein data themselves in a sort of an unbiased manner to find insights, new insights into different kinds of complex illnesses. So the example I often point towards is major depressive disorder, depression. We are currently writing up a paper where we've identified 3 different kinds of depression based on the proteomics, one that has a strong inflammatory component, one that has a metabolic component, and one that seems to involve disruptions to synapses and neurons. That could potentially lead to new and tailored treatments for depression, pending some further analyses. You can imagine a world where you recruit into your clinical trial based on an underlying proteomic signature and not just a clinical signature.
So in principle, I can absolutely see the trajectory of improving clinical trial success. And I'm excited to see, once we've had these data around a while, what the actual impact is. Yeah, thank you.
Yeah, I mean, that brings to mind a question I think for both you, Chris, and Rory. Rory, as a cardiologist, right? Cardiovascular epidemiologist and someone who has spent time, in the world of caring for patients and individuals. And Chris is a very entrepreneurial thinker in this space who's had firsthand experience with these data. What do you think are the most exciting near future possibilities for clinical impact?
Well, I think it comes to the right person point that Chris has made. And he gave a beautiful example there of depression. So there's the right treatment for the right person. And if there are more than one type of depression with more than one kind of pathway, then the idea that you would use a specific treatment for a specific subtype, I think is exciting. That probably will take some time before you get treatments that are specific for particular subtypes. Where I can see very rapid emergence of value from the proteomic data is coming back to this right person. Can we identify the people who are at risk of developing a disease much more precisely than we do at the present time? Can we use the proteomic data combined with other data to identify the people who will develop a disease and therefore be able to intervene with treatments we already have in a focused way, but early in their condition? And I think that may well be something that comes out of these data very rapidly and could be implemented very rapidly. Who should we be giving cholesterol-lowering drugs to? At the moment, we wait until they get to a certain age, pretty much, or we wait until they have a cardiovascular event. But could we use the genetic data and the proteomic data to identify the people who we should intervene in before their arteries flare up in order to avoid them ever getting to that point where they have an event?
It makes sense that the genetics and the polygenic risk scores are going to tell some of the story. I think proteins, as we've talked about, are catching additional information that are telling us about the person today.
Well, they combine the genetics, the lifestyle, the environment. They're pretty much, to a large extent, the common pathways.
Yeah, and we've seen lots of publications coming out recently with the first pilot data with polygenic risk scores, protein risk scores, and show that they complement each other, that they're really supportive of each other.
Yeah, no, Chris, I'm curious to get your perspectives and thoughts. I know that we've certainly had some conversations on the topic, and it's super exciting, seeing all the publications. But what are your thoughts on near-term possibilities and what could be tractable?
Yeah, I was going to say, you and I have talked for hours and hours over the phone and over a few beers on the topic of disease prediction. Is something we both are incredibly passionate about. And I do think, as Rory says, that we'll see the implications of those prediction tools, I would say, by the end of the decade. I think even shorter term, we'll probably see the most clinical uptake in the very short term in pharmaceutical trials. And I'll say that, I'll put my money where my mouth is. I think we're already doing this. We're already employing proteomics on trials to help better understand the impact of the drugs that we are, that we're putting through phase 1, phase 2, phase 3. I'm applying it in our neuroscience trials at J&J, showing how different drugs impact the blood proteome, with potential implications for repurposing and for drug filings. I think I just saw a paper published in Nature Medicine yesterday, which did this for semaglutide, showed the proteomic impact of semaglutide. So you'll see more and more of that over the coming years, I'm sure.
Yeah, and if I could just share from, my viewpoint, which is one of supporting a lot of scientists, both in the pharmaceutical space and in the, more traditionally academic research grant-driven space, there's a real coming together, merging is probably the better word, of these worlds, right? Where there's folks that have these beautifully characterized cohorts where if they have the access to population data from UK Biobank, they can then kind of hone in on a disease area of interest that they've spent perhaps a good chunk of their careers understanding, leverage proteomics, look at it in the context of a large population, and then there's often a triad of collaboration with drug development companies And I think that's a really powerful combination because you're lending someone's disease expertise that's bolstered with the weight of a population cohort. And then that can really inform far more efficient drug development decisions for folks, that see the value in this. So I can just share that I think that's incredibly exciting. It's happening today. And the next steps, I believe, are some version of risk scores and how they can be implemented in some way that's cost effective, convenient, and accessible to as much of the population as possible. I mean, that's certainly some time away, but I think it may come more quickly than people think.
We now have an amazing team that represents many aspects of Thermo Fisher Scientific, but what comes to mind is the complementarity of Olink, what Evan calls the next generation proteomic. Where does mass spec fit in, if you can share within that drug discovery pipeline for corroborating anything you're seeing in the UK Biobank data? Is there anything that you can share about that?
Yeah, certainly. I think mass spec is still viewed in many ways as the gold standard within pharma. We have a growing mass spec team at our J&J site in Cambridge, Massachusetts, led by Harris Beltaman. And in many ways, the mass spec sits alongside the affinity-based proteomics for discovery. We have an ongoing project for movement disorders where we are employing both affinity-based proteomics, Olink, as well as mass spectrometry to identify potential subtypes of movement disorders. And the data do very much complement each other. We see similar subtypes using both methods, but with the mass spec, you can often take it just that little bit step further, especially when you're using tissue like brain tissue. You can take a little bit step further, maybe go a little bit further looking at proteoforms, etc.
Well, I mean, I think along the lines of, where this is all going, I think another important piece of that puzzle is, to get the attention and capture the imagination of the general public outside of this, population research community, drug development community. I think there may have to be some sort of killer application or some sort of aha moment that raises people's awareness of the power and the potential impact of proteomics and how it could perhaps impact their own lives. I mean, Rory, as someone who's, spent as much time as anybody thinking of population epidemiology and the impact of the resource you and others have built in the UK, what do you think a killer app could be?
I always laugh about people's perception of health and the way in which medicine has gone. So, as someone who trained in cardiology and has been working in that area for a long time, I think the general public thinks, well, nothing much has happened. Except if you actually think back 40 years, we had nothing really that was useful for controlling blood pressure, for controlling cholesterol. You had a heart attack, you got into a coronary care unit, you were monitored and given some pain relief. The progress in the last 40 years has been phenomenal. And I think the general public doesn't really know that. And maybe that's the right way. Maybe the thing will be that what we need to do, as with genetics and with proteomics, is they just get incorporated into the system. We shouldn't be trying to train the public, or indeed most doctors, in genetics or proteomics or whatever. We need to build systems where it's like turning on the light. It just part of the standard things that happen. So I think the more invisible it is, the more likely it is to really change the way in which people are cared for, in which the NHS works. We will provide better care more precisely. It will be precise population health. We will be ensuring that we've identified the people who are at risk well before they develop the disease. We will have the kinds of treatments that Chris is talking about that are specific for the condition they are going to develop. And we will be able to implement those treatments in a more precise way for the individuals who will benefit from them. And the more that is kind of like turning on a tap, like turning on the electricity, like going to the television, the better, the more it is success.
So it sounds like integrated, woven throughout what healthcare will be in the future is the killer app. So woven through, the ability to understand what proteins are doing, woven through our predictive capabilities, and woven through improved clinical trials is the way to really make the biggest impact. Is that fair to say, Rory?
Yeah, I have no idea how the internet works. It just works. People use it. And that's what you want. You want this stuff. You want genetics and proteomics to not be cutting edge, but just the things that happen. And if we can make it like that, then I think health services will function so much better, and our governments will get better bang for their bucks. Our patients, the public, will get better health.
I think Rory's answer was excellent. I will say, for folks like us, sort of nerdy folks, super passionate about proteomics, maybe the proteomics equivalent of the folks that were in chat rooms on the internet in 1993, right? Like, we'll probably be looking for more subtle signs or a more subtle aha moment. I think there might be two. All right, I'll pose two different scenarios. I think the first scenario will be one in which we can unequivocally show that proteomics saves millions of dollars in healthcare and drug development costs longer term. It's still, I shouldn't really pose this to Olink, but it's still a relatively expensive technology to implement a higher throughput. So we need to show that that expense pays off, and whether it's through reducing the time it gets to phase 3, reducing the number of patients we need for a trial, or increasing the likelihood that a drug candidate actually will turn into a successful treatment, we just need to show that proteomics saves money. Or the second and maybe more powerful example is if we can show that proteomics saves lives. So maybe somebody discovers stage 1 cancer using a proteomic test and gets treated early enough to go into complete remission. And that detection of stage 1 cancer was impossible through any other means but a proteomic test. Or maybe proteogenomic modeling that identifies a drug target that turns into a cure for a disease like multiple sclerosis. Perhaps maybe somebody's misdiagnosed with a disease like Parkinson's disease. Maybe they have Lewy body dementia, and a proteomic test can show that actually they have a wrong diagnosis. It's Lewy body dementia, and they should be on this treatment instead of this treatment. So I think we will get there. I think proteomics can and will save lives, and when that happens, it'll finally be mainstream.
I love it. So, Chris, I love how you just have really been thinking about these things so clearly. You're so succinct in how you summarize the impact you expect in the future. So I'd like to kind of wind up and start with you, Chris, is if there were no resource limitations and the UK Biobank Pharma Proteomics project has been run on the full UK Biobank with the longitudinal representation in there, imagine a time in the future and it's exceeded all your expectations. No resource limitation. What do you imagine where you're sitting today that you would want to enable next?
I promise I will answer the question, but I'm going to take 30 seconds to just give Rory and Mark and Nye and the whole UK Biobank team some credit. I think it's already a world-class cohort, and I don't think proteomics at this unprecedented scale could happen in any other population biobank. And they've enabled that kind of an innovation by encouraging open access, by embracing pharma collaborations, and by really just incorporating this multimodal framework that I still believe is unparalleled. I don't know of any other studies that have 80,000 MRI scans. It's phenomenal. As somebody who, I did my postdoc with MRI scans with the Enigma Consortium, and at that time we were stitching together scans from different labs around the world, and now there's this one study from 3 different sites across the UK, all with the same scanner. It's mind-boggling. So it's a really hard act to follow. I think that the Beatles have already left the stage, right? So you're going to need the Stones and Queen and Led Zeppelin and some Frankenstein. Put them on stage and you might stand a chance of following up successfully. So I guess in biobank terms, right, I think that Frankenstein, that would probably be a cohort that already has the open access model of UK Biobank. It already has the longitudinal design, the large collection of multimodal data that I mentioned, including those MRI scans. But maybe in addition, you could add maybe recruitment of more non-white participants. I think at the time of recruitment for UKB, it was very representative of the UK population, but maybe increasing the non-white participants could be useful. The ability to recall participants for clinical trials could be useful, and maybe the integration. This is more of a sort of a pipe dream because it's very specific, but the integration of more specialized clinical scales for someone who works in neuroscience. I'd love to see the Unified Parkinson's Disease Rating Scale, UPDRS, or maybe the Hamilton Scale for depression, things like that. So
Fantastic. And Rory, no resource limitation, exceeded all your expectations. What's next for the UK Biobank or healthcare as a epidemiologist?
Well, the great thing about being involved in UK Biobank is that my expectations have always been exceeded by the way in which the scientists around the world have used the data. And I mean, that was what the Wellcome Trust and the MRC wanted. They wanted the data to be used by as many different imaginations as possible. And I think that has been really exciting to watch, just how different people have approached the same data in different ways and discovered really interestingly different things. But we focused a lot on the baseline samples, the samples stored from 20 years ago. I think that, as Chris said, the repeat samples being combined with imaging is very interesting. But I think also what will be interesting is the change from baseline to that repeat sample and changes in proteomic data and how that predicts disease subsequently in the longer term. We will have that repeat data on 100,000 or so people who've come to our imaging assessments. But I think what we should be trying to do is getting repeat samples on the whole of the cohort. Because my view is that where proteomic measures from 20 years ago are likely to be very strongly predictive of disease, changes in proteomic measures are likely to be even more strongly predictive and more specifically predictive of particular diseases. And the cohort is now maturing. So what I would like to see is getting all of the cohort back, getting biological samples from all of them, assessing all of them in terms of their frailty and their aging, so that one could look to see how do the baseline samples relate to aging processes in all of the participants, but then look to see how the changes in the proteomic data between baseline and, say, now are associated with development of disease in the next 5, 10, 15, 20 years. And I think change in proteomics, unlike genomics, is going to be a massively powerful source of information.
I'll also add to, what I think the UK Biobank has done exceptionally well is created an environment of trust with the participants.
The altruism of the 500,000 UK Biobank participants is unbelievable. I mean, their trust is really critical and something that we take very, very seriously. But their altruism is extraordinary. The fact, to Chris's point, that 100,000 of them have been willing to travel up to 100 miles, spend 5 hours going through an imaging visit, and then 60 to 70% of them are willing to come and do it again is unbelievable.
It's amazing.
I think the UK Biobank participants are the ones who really deserve all of our respect and all of our gratitude for what they're doing for health around the world.
Rory put it very, very well that we should all be grateful for the resource and the altruism that's enabled it with UKB. And I think in the course of this discussion, I'm struck by perhaps two core points of impact on the world that this work can have. One of them is, as Chris mentioned, precision medicine, right drug, right patient. And that's along the lines of a disease endotype exercise, where if you can get enough data on enough people, there's probably more than just one or two kinds of Alzheimer's, right? There's probably lots of subtypes for all of these common diseases that are creating real societal challenge, and dissecting those differences and eventually coming up with treatments to address those differences, it will have incredible impact. So that's one vector that I'm very excited about. And I think, to really advance that, we have to keep doing more. We need more cohorts. You need volume. You need N, right? We need a lot of patients to be analyzed. But the real power of proteomics is in its dynamic nature. And the longitudinality that we're going to get a really nice taste of from the full project here, I think will point very clearly that perturbational cohorts that do have multiple time points over as long a period as is feasible, will really start to help us understand the proteins whose dynamics are really important for diagnostic purposes. So I think we need to do a lot more of everything. And I'm not just saying that because I work for a company that supports that. I just think we really, as a community, will benefit across multiple vectors, but by continuing this work. And on a personal level, I'm committed to supporting, the kind of innovation that John Heimer did to try to make things happen, irrespective of commercial gain. And I hope that we can continue partnerships like the one we've built, and the friendships and relationships that I've built with Chris and Rory. I mean, that's the kind of stuff that matters far more than everything else. So,
yeah. An opportunity of a lifetime. It really is such a privilege. We're very fortunate.
And John definitely deserves his dues. John Heimer, he really made this happen. But so did you, Evan. You've been instrumental. You mentioned when we had lunch, and I'd had that vision to conduct proteomics since maybe 2016 in a cohort like UK Biobank, but it wasn't until I met Evan the following year that it became a reality. I think I came to him with that idea, and others had maybe dismissed it slightly or derided it, but he listened and he believed in it. He shared it, and he moved mountains at Olink to make it happen. So, thanks, Evan.
Thank you, Chris. Very kind.
Awesome. So, speaking of sort of what's next in terms of cohorts, I'll just make a call out to those who are listening to this podcast that Olink has an absolute passion, commitment, excitement around the matchmaking function of being able to bring cohorts to our pharma partners, bringing those to our nonprofit partners, to biotech partners, those folks who are in search of the right samples to demonstrate an understanding of various diseases. And so I think we need more cohorts. We need an understanding of the value and the uniqueness of all of the cohorts that we can connect you. We can build those connections, because that's, I think, really an opportunity to bring people together. With that, I will say thank you all for being here to talk about this phenomenal international resource that many folks are querying over and over again to build an understanding of insights over time. Serantis will be back with us next time. And with that, I will bring this episode of Proteomics in Proximity to a close. Thank you.
Well, that wraps up this episode of Proteomics in Proximity. Huge thanks to our guests and authors of such impactful publications. I also want to thank you for tuning in. Really appreciate you being here. If you enjoyed the content of this episode, please think about sharing it with friends or colleagues you think might be interested in the content. In addition, if you'd be willing to head over to Apple or Spotify or wherever you digest your podcasts and give us a rating and review, this will help others find the podcast when they're searching for proteomics or precision medicine podcasts. And mostly, I want to say we would love to hear from you. So we have a dedicated email address, pip@olink.com. Please reach out. Let us know what you're interested in hearing about, what you care about, and any feedback on the episodes that we have already done so far. This is all about you, and so we're really keen to make sure that we're meeting what you'd like to hear about. Thank you so much, and we'll see you soon.